Thank you very much for Chairman for nice introduction. Today, I would like to talk about these topics. My name is Taguchi. I came from Department of Physics University, Tokyo, Japan, and this is collaborative work with Professor Taki from Saudi Arabia. The content of this talk has already been published as preprint here. If people are interested in the content of this talk, please check this print after the conference. So, introduction. Usually, transcription cannot be cooperate with DNA replication because RNA cannot be transcribed from DNA during the DNA replication take place. Thus, once the DNA replication process started, transcription must be terminated. But if every time transcription process has been terminated, the genome state, for example, transcription factor bindings or histone modification is reset, not replicated to the DNA. So it is very ineffective. So the status of genome during RNA transcription should be bookmarked such that it is quickly recovered after the DNA replication has been finished. But how? Today, I would like to talk about the bookmark mechanism of this process. I have analyzed this dataset. This dataset formatted as tensor XIJKMS and with this number of dimensions. And I stands for genomic region. Each genomic region is length of 25,000 base pairs, and whole genome divided into this length of regions. And there are 2 cell lines in this experiment, and 3 kinds of histone modification are measured, and number 4 is input. This is some kind of control. And the measurement has been taken place the 3 cell cycle. And for these observations, there are 2 biological replicators. And the tensor has been decomposed using Tucker decomposition, employing higher order singular value decompositions. Tensor defined in the previous slide has been decomposed into multiplication of smaller core tensor and 5 singular value matrix. So there are 2 other singular value matrix, but I cannot draw 5 dimensions, so they are omitted. So mathematically, this tensor decomposition can be written like that. Here the vectors with attributed cell lines, histone modifications, cell cycle and replicate, and genomic regions. The weight G is the weight of the summation of this product. Then the process, we can select genomic regions, which is associated with reactivation is as follows. First, we try to check which vector attributed cell cycle represent reactivations. Next, we try to which vector attributed histone modification associated with distinction between control and histone modification. And for cell lines, the biological replicators, we try to find there's a difference between cell lines and replicators. And considering with these 4 vectors, we check the weight of G such that having a larger weight with these 4 vectors for genomic regions. Then we have found which singular value vector are used for genomic region selections. So this is 3 singular value vectors attributed cell lines, cell cycles. So it is clearly third one is once it's down and up. So this is activation process. So we have employed this reactivated process to be used for further analysis. Then this is the 4-vector attributed to the histone modifications. And this letter O represents the control. So this means there are no difference between histone modification controls. So this vector should be omitted. So other 3 vectors has, in some sense, distinction between histone modification and control. Thus, we have decided to employ these 3 vectors. For cellular in and biological replicated, we need to find no difference between the cellular ins and no differences replicated. So we have decided to employ the first vectors. Then we check the weight and fixing the first cellular in vectors and 2 to 4 histone modification vectors and the third cell cycle vectors and first biological replicated vectors, which L5 has having larger weight. Then we can find this one. Fourth vector has larger weight with other 4 vectors. Then the p-values attributed to the ith genomic region using this function. This is cumulative chi-square distributions. This means we have assumed U for I, which is attributed to genomic regions, assuming that always Gaussian. The p-value will be computed using cumulative chi-square distributions. Then 507 DNA regions are selected because their Benjamin Heisenberg criterion corrected p-values less than 0.01. Then we count how many gene symbols are included in the selected 507 DNA regions, and I've found 525 gene symbols. First, we check it is the selected 500 genes are really coincident with the reactivation or bookmark process. So they are a little bit busy tables. I have no time to explain it very clearly, but for this means it's 3K that 27AC is associated to bookmarks. These 2 histone modification is bookmark, and these 2 bookmark histone modifications are distinct between controls. So basically, 525 genes associated with histone modification coincident with reactivation or bookmark process. Then we like to check if the selected genes are associated with transcription factor bindings. This is the result of Enricher, which is a famous enrichment server, and here's a huge number of transcription factors detected that targeted selected 525 genes selected by us. And there are some transcription factors biologically important, included these 7 categories, this one, and these biologically important transcription factors are really included in our analysis. So our analysis was successful. The last test is if selected 525 transcription factors can work as bookmark TFs. So as you can see, top 10 transcription factor that target 525 genes over these 7 categories. Some of them are known to be bookmark transcription factor during a post-mitotic process. So I have no time to explain in details. The conclusion. Bookmark mechanism of reactivation of transcription after dereplication was studied. Then the TD-based unsupervised FE applied to the histone modification profiles before and after DNA replications. And some candidate bookmark tests were successfully identified. Thanks for your interest.